Alrighty guys, it's me, Olivia, your trusty TA. Today we're going to be taking a deeper dive into R, specifically to study what my master's research is in, which is the microbiome. And I'll show you how some initial steps, how we analyze that in R. Tutorial aims you'll be able to demonstrate basic analysis of microbiome data by the end of this. You'll determine how communities can be affected by different variables of interest. You'll be able to perform basic measurements to characterize microbial data. And you'll be able to link genomes to phenomes to better understand the physiology and leverage knowledge in collaborative research projects in the gut microbiome of herbivores. So I'll start off with a quick synopsis of what metagenomics is and what the microbiome is. So metagenomics is the study of genomes within genomes. So directly from environmental samples. In this example, we analyzed 16 srRNA sequences from a population of moose in order to profile the bacterial community associated with their gut. So in metagenomics, we care about the micro. We care about what are the bacteria doing in the environment. So there's soil metagenomic, there's a soil microbiome, an ocean microbiome, there's an atmospheric microbiome, there's even like as much like every environment you can think of, there's microorganisms in a microbiome. Inside of that, we have a gut microbiome, and the moose that we're studying are going to have a gut microbiome that we're going to be looking at today. So what this is based off is amplicon sequences, which means it's just only sequencing the RNA or ribosomal DNA of organisms. And the main questions we want to know are which bacteria, who's there, and in what proportion. So who's dominating, what's the structure, what's the overall community. So more background. So this is what like ribosomal, that's E. coli ribosome, right? So it's a ton of RNA, so you get a lot of information from these samples. It's really targeted so that you're only targeting specific regions of the ribosome. So in our example, the data set that we have today, it's a V4 through V6 region. I lied, V3 through V4. I almost forgot. V4 through V6 is another common one. But they target specific regions that they know have highly conserved regions, so that they also have huge reference databases. So this is like the regions that they'll target. There's regions that are super variable, so they know that they can reference them to the databases. But with amplicon data, we're mostly just looking at where the sequences are coming from, so that we can do what's called taxonomic assigning. So taxonomic assigning is when we classify the bacteria based on their RNA sequences. So that's limited in the microbiome because there's a big bulk of genomes, bacterial genomes, that have yet to be characterized. A lot of that has to do with like not being able to be grown in a lab. Some of them need other bacterial species at the same time to grow. Other ones need like aerobic environments, and you can't replicate that in the lab. So you'll see it today when we do the taxonomic assignments. It'll just say uncultured rumen bacteria, just because that's what they know about that. So it's obviously still a work in progress. So to kind of touch on what we're discussing today, so we have a population of moose in Michigan that have been studied over a 6-year period, and they're studied because of where they live. So they live on Isle Royale National Park in Michigan, which is a pretty secluded island. So you're kind of better able in Lake Superior, so you're better able to control more of the environmental variables. So fecal and urine samples were taking over a 6-year period, and essentially it's in order to study the role that plants produce as part of their normal metabolism and as part of a chemical defense, they produce what's called plant secondary metabolites, or compounds. These are toxic to a lot of the mammals that eat them, and that's by intentional design. The plants are trying to protect themselves, so they make these super tough compounds because they don't want to be eaten as much. And then so some animals, it's a co-evolutionary arms race, it's described. They're developing a way, animals just develop a way to continue to eat these plants without getting sick or having their detoxification process totally overloaded. So moose are one of those mammals that have been able to do that with balsam fir, which grows pretty abundantly on the island. Other plants are cedar. Cedar is shown to be pretty toxic to the moose. Here in Idaho, we have sagebrush everywhere. Sagebrush is actually pretty toxic to a lot of animals. It's not super nutritionally dense, so they're not getting a lot of nutrients from sagebrush, but it's like the only thing that grows in the winter. So pygmy rabbits, mule deer, that's kind of what they rely on. So the moose is a similar situation. Balsam fir is one of the only plants that grows in the winter, so it's kind of what these moose rely on. Some background information about the moose. The balsam fir has been diminishing in the western part of the island over time, and it's remained pretty stable on the eastern side of the island. So some initial hypotheses going into the project would be, okay, balsam fir is shown to have some toxic chemicals, toxic properties. If there's less on the west, then the moose on the west should theoretically have a higher bacterial diversity in their microbiome, right? Because it's not being depleted by toxic chemicals. That's at least initially what you might think, and you'll find out through your analysis today that might not be the case, and so you might have to think about why. So the aims of this, why we care about microbial research and microbial data. It can tell us a lot about the fitness of an animal, but it can also tell us a lot about communities as a whole. So if we see shifts in microbiomes that are consistent in multiple different organisms within the same population, we can make inferences about how their environment's affecting them and make community level inferences. The other thing is the human microbiome, right? That's a huge area of research now that it's shown the microbiome. The bacteria in your gut has been shown to be associated with immunity, with allergies, with even cognitive processes. So it's a huge area of research now, but that also means there's a ton to be discovered. We do know that the gut microbiome in mammals and in humans has a larger part to play in the health of the animal that we just had not previously thought before. So it is a pretty cool kind of new area of study, and it's really insightful, and you're learning a lot about how we are a process of multiple different we are a product of multiple different processes that happen within these microscopic organisms. It's kind of cool to think about. So today the tutorial will go over, I'll explain what kind of in detail different approaches we take for statistical analysis of microbiome. So overall, this is kind of a nice flow sheet. So in this situation, it's fecal samples. You send those off for sequencing, and then you get raw sequences back that are literally just like A, T, G, they're just in a row. And then you do what's called demultiplexing, and you join those sequences because they read them reverse and forward. And then, so you match those sequences to their sample, and then you create what's called a feature table or an OTU table, or now an ASV, amplicon sequence variant, where it's a table of how many different species are present in a set of samples. And from that table, you can then go on to do testing for diversity analysis. And then you'll also assign taxonomy. So that's the who's there. So you assign taxonomy, and taxonomy is kind of tough with the 16S because you're really, again, you're using reference databases, so it's only as good as your reference. And then you can build a phylogenetic tree. So some of the big questions that microbiome researchers ask are, how many different species are present in a set of samples? How much phylogenetic diversity is present in each sample? So within each individual moose, how much diversity do they have in their microbiome? How similar or different are each individual moose's microbiomes to the group or to one another? And then what factors are associated with shifts in microbiome composition? So pH, elevation, blood pressure, body site, host species. Today we'll talk about how region and location on the island has seen to be a shift in the microbiome. So at this point in the analysis, we're going to skip ahead a little bit because I think importing and checking data, we're going to step ahead just to step 4 super quick so I can just make sure that you've downloaded all the data. So first you're going to need to download the data. You're going to go to your Blackboard course site, go to Lab Modules, Mammal Microbiome, download all of the files in the folder titled Data. And just like you did last week, you're going to want to put those in a labeled folder. Whatever you want to name that folder, you want them all to be in the same place because we need to set our working directory for RStudio. So once you download all those files, then you'll have a feature table, which is a list of all the different types of species in your samples, taxonomy classification results, so who are they, phylogenetic tree, and a metadata file. So a metadata file is basically just like all the history about each sample. So the metadata file has like when the sample was collected, what the animal was eating at the time, the animal's age, whether it was a male or a female, where it was a juvenile or an adult. So it's just back history on the samples, which you can use in your analysis to test what factors are causing changes in the diversity. So we're going to start here. We're going to open our RStudio. So we'll come back to the PowerPoint. We're going to open RStudio. Oh man. Here. So when you open RStudio, I already have like the script loaded. So you'll click on File. Let's pretend you do it. File, New R Script. And then just a recap, this is from last week. You're going to download a lot more packages this week than you did last week. That's okay. So first, we always want to give descriptions of what we're doing. Start by writing so the date, the main goal, whatever, Olivia Rodriguez. I didn't even write the date. And then you're going to do what's called setting the seed. So this action allows you for reproducibility of your code. So a lot of the times in microbiome analysis, you're doing things where you're like, it's like randomized. And in order to keep that, to get the same results, you want to set your seed, and it's a 4-digit number that'll be saved, and it's not going to be saved when you leave R. So you want to make sure it's a 4-digit number you remember, and then that way somebody else could theoretically click run all on all of your code and get the exact same results. So once you do that, we're just going to go through and install packages. So if you remember from last week, the installing packages function is install.packages and then parentheses package name. And you can type that either in the console or up here in your actual script. It doesn't really matter if you type it in the script because you don't need to really save it. But again, once installing a package doesn't mean you load it. So once you install the package, still go back in and load all of the packages. So you're literally so we'll start that now. I've installed all the packages. So now I'm going to highlight all of these and click run. That loads all of the packages in my that loads all the packages so that they're ready to use, so that I can use the functions associated with these packages. Did I click run? Yeah, I clicked run. So then the next step is we're going to install, it's a special type of package. So you'll notice that the files that you downloaded end in a QZA. That goes back to the type of analysis I did. So I use something called Chime2. So Chime2 is basically, it's similar to R. It's its own language. It uses like Conda, but it's specific for microbiome research. So it's just the functions are specific for microbiome research. So that's where I processed all this data, and then I like exported it, and I'm using R to transform it all. So just because R is typically better for like visuals. So that's the why it ends in that QZA. So you're going to need to install a special package, and you'll need to do this code exactly. And then once you install that package, still remember to load the package. And then once we do that, let's make sure we have our working directory going well. So if you remember, there's two ways that you can check on your working directory. To see where it is right now, you can run this code, get working directory. So when I ran it, that's my working directory points to a folder on my desktop named Microbiome Tutorial. And if I want to set working directory, you can either type it, or you can go to Session, Set Working Directory, choose Directory, and then you choose the folder. So if you downloaded all your stuff in a specific folder, make sure you're choosing that folder. Okay, and then so next we're going to import and check our data. So I talked about this. This is just background on where the data comes from. It's fecal bacterial microbe data. So we're looking at variables like region and year. And again, this is where you're getting your data from the Blackboard course site. And then you're just going to follow these steps to import your data. So today it's easier if if I don't type them out, if I just do them as in real time, just because it's like there's a lot more code and it would just take forever. So this first line reads in the feature table, and I'm calling it SVs. And then I'm going to change it to an actual table instead of a matrix. Well, right now it's a list, so it's a matrix, or it's a list. I'm going to change it to a matrix. And then I'm going to assign the column names so that they match up appropriately. Oh, whoops. I didn't run this next one. Whoops. Now I changed it to a table. So now it's a data frame. You can see it there. And now I'll match the column names so they match up. So then if I click on this, I should see a table. Which I do. Perfect. So this is just like that. So that Chime2, this is the unique codes they use for taxonomy. So you could just ignore this for now. Now we're going to read in our phylogenetic tree. So same thing. I'm going to name it tree. And then I'm going to turn the tree into a matrix. And now we're going to read in the metadata, which is the history about the sample. And then this row names, I'm making sure they match appropriately. And now we're going to read in taxonomy. So the taxonomy will read in as like a list. And then if you look at it, you should see like, oh, so here's the feature ID, the specific unique ID. And here's the taxon and the confidence. It reads in as like a string, but you want to break that up to make sure it's reading as like a table. So the way you do that, it's called the parse function, parse taxonomy. So you do that, and then you click head again, and you do head again. Now it reads like a table. Which is what you want. And then all of these so this as.character, if you remember from last week, or from last lab, you'll notice that like these are being read in as as factors and I need them to be read in as characters. So I just go in and change literally every single one. And then I turn it into a matrix. And now we're going to create what's called a phyloseq object. So the phyloseq object is basically a cool way in R to combine your phylogenetic tree, your sequencing, your sample data into one whole object that you can just call upon to do downstream like analysis. So it's what we're going to use for the remainder of the tutorial. Basically, like you're calling upon the object every time, so it's vital to be comfortable with the general structure of this object in order to understand it. So after you create the phyloseq object, you want to summarize it. So we'll follow this line here to create the phyloseq object, and then we'll summarize it. And the phyloseq object does exactly what I said. It creates an object that combines the table, the sample data, the taxonomy, and the phylogenetic tree. And it tells you, hey, this is how many samples you have, this is how many variables you have, this is how many taxonomic ranks you have, this is how many tips and internal nodes your tree has. Which is just good to know, so it's a good way for you to double check. You're like, hey, 324 samples, I thought I had 400 samples, what happened? So it's a good way for you to go back and double check that. This next step 6, you're going to skip it for now. This is part of your deliverable. I left it in the protocol so that there would be like instructions on how to do it, but we're going to skip it for now while we do the protocol. And you're just going to move on to this step here, which just creates a sample metadata data frame. So now we're going to go on to what's called diversity analysis. So I'm going to take a back pedal and go back to the PowerPoint. So in microbiome, in the world of microbiomes, diversity is characterized by two things, alpha diversity and beta diversity. So what that means is alpha diversity is the diversity in a single sample. So in just one moose, what's the bacterial diversity? And there's two ways that you classify alpha diversity. How rich the sample is and how diverse it is. So richness describes how many different bacterial species are there, and diversity describes how well are they spread apart. So in microbiome, like being rich is good and being diverse is good. So you want ideally you want high richness and high diversity, or high evenness. Evenness is another way to say diversity. Oh no, it's not. Evenness is just like how ideally they're not all the same, but you want high diversity and high richness, because you want to be able to say there's a ton of different people there, and it's not just there's a ton of bacteria and it's all the same one, right? Because that's actually unhealthy for an animal to have a low level of evenness or a low level of richness. So here's a good example to explain that. Excuse me, I don't know why I keep yawning. So in this first, you have low richness and low evenness because you only have 6 pieces of fruit, and 4 out of those 6 pieces are apples. So it's apples dominated, and it's not really that much fruit. But then on the other side of the spectrum, you have high richness and high evenness because you have 7 different fruits, and it's totally varied. I guess bananas is dominating a little bit, but it's pretty similar abundance to of most of everything else. So that's alpha diversity, and there's a few different tests. So for richness, you use ACE or Cal1. For diversity, you use Shannon's or Simpson's. With Shannon's, the higher the value, the greater the diversity. And then you can also do phylogenetic diversity, where they sum up like the taxonomy and the phylogenetic relationship when assessing bacterial diversity. And then you have what's called beta diversity. So beta diversity is differences between groups, so between communities. So it's between sample diversity. So how different is each sample from every other sample? So not how different is just one sample, how am I, like, how is my bacterial diversity, but beta diversity is how do I compare to you and that person and that person and that person. So with beta diversity, there's two types. Again, there's two different ways you look at it. It's either abundance or presence and absence. So abundance is diversity, presence and absence would be like richness. So here's a good chart to explain the difference between like alpha diversity and beta diversity. So with alpha diversity, you have 3 different types of goldfish in site A. So that's the alpha diversity for site A. The beta diversity between A and B is 2. The beta diversity between A and B is 2. Because here, and then the same thing, the beta diversity between B and C is 3. So, but then here you have the alpha diversity, you have 3 different types of fish. Here you only have 2 different types of fish. So, 3, and then you just do the math, right? So 3 minus 1 and 2 minus 1, and then your beta diversity is 3. So it's how different is B? So site B has 2 different values for beta diversity. How different is B from A? How different is B from C? So then now that we know, another good, it's in the actual tutorial to explain the difference between like evenness and richness. It's a great one with the trees. So like community 2 is actually the worst community because they're totally dominated by species A, these trees. So that would be a community that like might be, their microbiome is not as great, and this is actually a better microbiome. If we're talking like in terms of like bacterial species. So alpha diversity is kind of usually the first thing you do in microbiome pipeline. We're going to focus on Shannon diversity, which is the overall community diversity. It measures richness. And then we'll also do a Cal1s, which again, just Cal1 just measures richness, but in a different way. Shannon more measures diversity. So then beta diversity, we're going to look at the Bray-Curtis. So that was that abundance diversity. And then that's going to be the only one we actually look at for Bray-Curtis. So it's a quantitative measure of dissimilarity. So that's really what you're testing for in beta diversity, is how dissimilar are 2 communities from one another. So if you're following along in the tutorial, our phyloseq object, we named it phyloseq 16S moose, is ready for analysis for alpha and beta diversity, and we're going to start with alpha diversity. So the first thing we're going to do is we're going to create we're going to create just another like data frame or table, but that has all alpha diversity values. So there's a cool option that's just estimate richness, and then we're going to call it alpha, and that's going to give us all of the measurements of alpha diversity for our samples. So if we run that now, so alpha, then I should see it pop up. There it goes, right there. So that's again a data table. And then I have to change the row names to make sure the row names in this data table match the row names in my metadata table, which is that like history table about the samples. And then I have to make sure the sample ID is at the start. And then left joining is just, all I'm doing is attaching the values of that alpha table to the back end of my metadata table. So we can look at it now because I called it alpha_meta. So it's going to be this huge thing. So see columns 1 through 50. If I go all the way this way and I scroll to the right, see now I have all these alpha diversity values. I have observed OTUs, Cal1s, Shannon, Simpsons, inverse Simpsons, and this joining by sample ID allowed me to tag that on at the end. So now that we have that, I can plot stuff. So the first thing we're going to do is we're going to plot the species richness for Cal 1s and Shannons, and we're going to color it by year. So when you do that, so it takes a little minute when you create these plots, you should see a plot that comes up like that. So here's Cal 1s, here's Shannons, and it doesn't really look like there's a huge difference in Cal 1s, which is evenness. I mean, you can see like maybe the West seems to be, by year, seems to be spread out more. But Shannon's diversity, the East has an overall higher Shannon's diversity than the West does. So that's that within sample diversity. What that's saying is individuals in the East have a higher bacterial diversity than individuals in the West. With higher individual bacterial diversity. And then we'll do the same thing for location. Now we're going to, the x-axis is going to be year and the coloring is going to be by location. It doesn't really change much, it just kind of affirms what we know. So again, Cal 1s and Shannons, and then I colored by location. So you'll see, you'll just notice overall the pink is higher than the blue, and you do have some outliers here and there, but overall the general trend is the Shannon's diversity is higher in the East than in the West. So alpha diversity is higher in the East than in the West. And then this is the plot that we're going to create, the kind of overarching plot. So it's just Shannon's. And this kind of, again, affirms what we already saw. Here's the East on the x-axis, here's the West, here's it colored by year. And aside from a few outliers, it looks like overall the East side has a higher Shannon diversity than the West side. Or individuals in the East have a higher Shannon diversity than individuals in the West. So then again, remember, if you want to save those, you can just save it. Or you can go to Zoom and save it that way. Now we're going to go on to beta diversity measures. So the first thing we got to do is we got to transform the data to where it's in a compositional format, so what that means is microbiome data is kind of hard because it's all relative to one another. It's all within a piece of the pie. So compositional allows you to kind of do that absence or presence testing, while also maintaining the fact that it is all relative to one another. And then we're going to create what's called an ordination of a PCOA. So it's a principal component analysis. And it's a way to structure the ordination that we created so that you can see, it's a way to structure that composition, because beta diversity is hard, right? Because every sample, so there's 324 samples. So each sample has 323 values attached to it because it's how different each sample is from every single other sample. So this ordination basically creates this distance matrix that you can quantify. And now we're going to plot it. So I added a couple specifiers on here. So you would just have to, all of these are just specifying what I want the graph to look like. And now print is going to show me the graph. Okay, so the way you analyze a PCOA graph is by clustering. Oh, why I keep yawning? It's by clustering. So you want to look, so typically they're actually in 3D and you can move them around to see how they cluster. You want to look for distinct clustering because then that tells you that you have two distinct groups. That if you group them by location, you see two distinct groups based on their microbiome, based on their bacterial diversity in their microbiome. And here I would say I kind of do see two distinct groups because it seems like the pink are kind of clustering together over here and the green kind of clusters together over here, aside from, again, those few outliers. So basically what that's telling me when they cluster together, when they cluster and it looks like they're clustering separately, like if the circles were right on top of each other, then you would say, no, you don't have two separate groups when you group them by location based on their bacteria. They look the same. What this is telling me is that like the bacterial diversities in these two subpopulations, the moose on the east, the moose on the west, does differ. The overall compositions are differing from each one. Okay, and then the last two graphs we're going to be creating is what's called, I'll scroll down, I'll keep going, whoops, is just kind of like an exploratory thing. It's looking at the taxonomy. So that's, again, naming the bacteria that are in the sample. So not just saying, hey, the bacteria analysis showed that they were different. It's saying, who is that? So the first thing we want to do is remove anything that's not bacteria. So we're going to filter our data set for specifically things that are in the kingdom bacteria. Oh, whoops, I forgot to run the whole line. There we go. So specifically, so that way, I mean, I think there was some archaea in there, but we want just what's in the kingdom bacteria, because that's what we're looking at, bacteria. So now the next thing we're going to do, we're just going to create an initial plot, just looking at all of the bacterial families that were characterized by our reference databases. This one is going to take a little bit on your machine because there's a lot of families. That's okay. It's just going to take a little bit. So you just wait, or you make sure you understand what's happening with the data. But this one should, oh my goodness, why do I keep yawning? I'm so sorry. I ran it several times, and it works. It just takes a minute, and you'll see why, because this plot is kind of big. So if you scroll down in the tutorial, you can see all of these different families that are characterized. So it just takes a while for R to kind of build those bar plots. So don't get worried if like you see that the stop sign has stopped and you don't see a plot. Just give it a minute. Just give it a minute. It takes a little bit. Yeah, so there it goes right there. And the easiest way probably to look at this is I just hit zoom, and then again, just to give it a minute, because it's basically like it has to load the graphics is honestly I think like what takes forever. And it doesn't help that I have like a million tabs open all of the time. But okay, to save us time, when you do open up that graphic, you should see something that looks like this. And when it finally loads, and what this is showing you is location east, location west, is the abundance of bacterial families. So it groups them by abundance, from high abundance to low abundance, from east to west. So you can see whether or not they differ or they stay the same. And overall, you see like in the east, it just seems like they seem to have more of a bacterial load entirely than the west, because they have abundances that go past this mark here, where as the west barely does. So they're past this mark here, the west doesn't, the east does here. So it's a good assessment. It's kind of like, sometimes people do this actually at the beginning, so they can just say like, okay, what am I working with here? What families are the most prominent seen in my bacterial data set? And this is where I was saying like, those genomes that have yet to be characterized, uncultured. So unidentified rumen bacterium. So that's just kind of like placeholder names until someone can go out there and characterize them. Okay, so now after we did that, there's a ton of families. So we want to let's just pick the top 10 most abundant families and just look at those. So we're going to call those top 10 F names. And we're going to sort again our phyloseq object for the top 10, families 1 through 10, family. Taxa table is the taxonomic table, and we're looking for just families in the top 10 families. And when we run that, I'll run it here on mine. And then once you do that, you want to take out anything that's not in the top 10 families. And then once the stop sign goes away, you should just be able to plot that. And like last time, it'll take a minute. You just wait. While you're waiting, you can also start looking at the deliverable and seeing what the requirements are for you there. But when it finally does load, you should get an image that looks like this. So this is actually pretty cool because this graphic shows you all the top 10 families. It puts the legends are right here. And then it shows you how abundant they are, east versus west. So the two most abundant families are obviously the Lachnospiraceae, there it goes, Lachnospiraceae and Ruminococcae. And then that's followed Bacteroidetes. And these are actually two bacterial families that are pretty prominent in us as well. And then you can look at the ones that are not as prominent. I guess it's that one. But like for whatever reason, this Barnsillaceae way dropped down in the west when compared to the east. So that's also another thing that, like, if you're a researcher and you're doing more research on this, you would go and look at, like, okay, what's going on with this specific one? You could do another bar plot. Okay, so this last step in the tutorial is totally optional. It's woo. Oh my gosh. It's just how you would export stuff and save it. So you don't have to do it. It's optional. It's just good to keep that in there if you're someone who's going to work with R. This is just how you export stuff and save it. So now let me talk about the deliverable, which means I got to go back to step 6. Which means I actually got to find it. Okay, so the on your own. Essentially here, you're going to pick 1 year. So this is the example for if you would have picked 2013. And you're going to do every step after this line just for the year 2013. So this line of code should no longer say... so now you're like, so here, if you're doing 2013, you shouldn't be writing moose there. You'd be writing 2013. And then, so you're still following all the same lines, but all you're basically doing is changing the moose to whatever year you chose. And again, the code to do that, to choose a year, subset samples, year equals 2013. So the only option you can't choose is 2016, just because they didn't collect that year. So let me open the deliverable. So I think I also, okay. So yeah, refer back to step 6 on your own in the mammal microbiome tutorial document. You will perform the same data analysis you did in the tutorial on a subsetted portion of the larger data set. So you will filter or subset the data of 6 years and perform diversity analysis and create these taxa plots on just samples from that year alone. And then the first 4 questions are basically you just attaching all your plots that you made. And again, adding figure captions and titles. And then there's a couple discussion questions. So the first one is, which region, east versus west, had a higher Shannon's diversity value? What does that indicate? Did the moose appear to have significant differences in beta diversity when grouped by location? Either way, explain why you think yes or no. And then what does the Bray-Curtis test for? Bray-Curtis test for. So the discussion questions, I mean, so it's overall, it's like 6 questions. Honestly, like, if you're following the tutorial pretty diligently and reading, it shouldn't be super hard. It just might take you a while to, like, work out kinks, you know, make sure you're not doing spelling mistakes and stuff. And then, like I said, these graphs take a little bit to load, so when they finally do load, I would just do, son of a nutcracker, I would just do export as PDF and then save it as like an 11x4, because it's just like no time to like go back and do them. And then once you do that, you have just analyzed the microbiome. You did alpha diversity, you did beta diversity, and you can tell me whether or not you think diet is playing a role in these moose. And that is it. So I'll just discuss where you'll turn stuff in on Blackboard. Son of a nutcracker. Okay, so if you go to Blackboard, you should see something called Microbiome Lab Module Deliverable. That's where you'll download the deliverable. This is where the PowerPoint is. This is where you'll get all the data. This is where the tutorial is. Yeah, I don't think there's anything missing. And then my Zoom office hours, I'll be in, I won't do the 2 to 4, I'll do the 3 to 5 today, because I can't do the morning today, and then Friday 10 to 12. So work through it, and if your graphs are taking forever to load, definitely let me know. It'll probably, honestly, once you subset your data to just include 1 year, I bet you they'll load faster. It's just because there's so many samples. But that is it. That is all I have for you today. Thank you guys. Have a good rest of your week. Bye.